Okay, 1 November 2012, Biology of Fishes again. Talking last time about the perception of environment and the fact that fishes learn about environment, use that information to organize their behavior, to distribute themselves, hopefully in places where things are better, if such places exist, and to avoid adverse situations. Before I move ahead and pick up where we left off last time, I'd like to make a couple of comments about lab issues. I just sent out an email to the graduate students, 617 students, saying that there is a project idea presentation posted on 7 April of 2011, and I'm going to convert that AVI to a MOV and then repost right away so that you can take a look at what I have suggested that people consider doing for projects in previous years. And some of those projects have been done, some of them haven't. As far as you're concerned, it won't matter. They'll be new to you. I've mentioned project ideas as we've gone along this semester. Again, some of them have been done already and some of them haven't. And every year I get a new suggestion from somebody. Somebody comes up with a novel idea that I haven't thought about. And if possible, we will work together to modify EcoFish and make it possible for you to evaluate that idea using simulation. So anyhow, I'm anxious to work with you on that. Zoe came up just before class started in the local group here and pointed out that the link for the data template for Lab 5 isn't working. And I think all I need to do is go in and change the link and it'll work. But I think what it was, Zoe, is I didn't remember exactly how I did that, and I wanted to go back and look at my own help files and make sure that I was going to be putting up the right the template with the right information in it. Obviously, in this case, all the data that you use are going to be your own data. So you'll need to replace the data that are in the template with your own data, and I got to make sure how that's set up and whether it's going to be a problem or not. Hopefully it won't be a problem. So I appreciate the fact that you and several other people are already moving ahead and trying to take care of Lab 5 and get that done. Thinking about Lab 6 and the project in the case of graduate students. So let me go ahead now to the point where we ended. I guess I like to always back up at least one step and sort of remind myself of where we were and what I was talking about at the end of the class last time. And we were talking about this idea of thermal lag being used by animals in general and by fish in particular to detect the fact that there is a change in environmental conditions over time. And I stress the fact that this is a time issue, a temporal issue, and not a spatial issue. But a lot of times, you know, if you're moving in space and there are gradients of environment in space or discontinuities, then those spatial gradients or discontinuities are logically translated from the animal's point of view into a temporal change, because you can't be two places at once, at least in the non-relativistic world. So that means time and space sort of trade places. So this idea came from work on tunas, and it's been published. And basically it says that fish might use the lag in their core deep body temperature relative to the superficial information they're getting from surface sensors to not only detect the fact that there is a change through time, which probably is related to a change in space. Maybe. If there isn't a change in space and the animal can't do anything about it, you know, things are going to go on in time, it's not going to control time. But if the change is because there's a spatial distribution, a variation in the spatial distribution of something, then the animal might be able to use that information to better its situation in the next moment. by doing the right things in response to the information. And so the idea is that if you're headed into a future where temperature is going down, or at least has gone down in the recent past, then the core value of that temperature would presumably lag behind, and the more thermal inertia you have, that is, the larger body mass relative to surface, the presence of countercurrent exchangers that retard the flux of heat, the more of that you got working for you, the faster you move through space to maximize the gradient, or to maximize the variation of the factor over distance and time, the more this differential. And so it might be that fish use that information to inform themselves physiologically about changing thermal conditions. If they're going into a cooler world, then core temperature is going to be warmer at every moment. And into a warming world, then core temperature is going to be cooler. And you can even calculate the maximum value of the difference at steady state, the differential between TB and TA once they start moving at the same pace. That's going to be dTA dt over K, where K is the coefficient for heat exchange. And you can see why then tunas should be better at this than ordinary fish. And that's why we got the idea with tunas. So, does Ecofish use this? No. Ecofish doesn't do it this way. Ecofish is using temporal changes in metabolic scope for growth. I originally programmed Ecofish to use this thermal lag hypothesis, but then I got interested in the notion that it might be possible to optimize total environment by maximizing metabolic scope for growth. And so I decided to go ahead and do it that way instead of this way. But if you're talking specifically temperature, then this is an idea. And there's some experimental data coming from very clever experiments done by this guy, Larry Crawshaw, that sort of pointed us in that direction, or were done at about the same time, actually, as we were getting these ideas. And what he did was put little brown bullheads in longitudinal gradient tanks, and with temperatures that were from 9 to 30 degrees in this particular example. And then the thermode was a device, basically it was two hypodermic needles, one inside the other, fine, gauge, with the end on the outer needle plugged or welded shut so that a fluid could be put down the inside hypodermic needle and in return flowing around the outside of that needle and inside the bigger one, as you can see here. In other words, you can see that this would be a one-way street where you could put in a fluid and then let it return out of the fish. And he was implanting this in various places within the fish. And he was controlling the temperature of the fluid so that it was relatively warm or relatively cold compared to the ambient temperature of the gradient where the fish was. And he got some very interesting results. If he put it in the anterior forebrain, the frontal lobe of this little catfish, and had the temperature of the fluid that was flowing at levels from 23 to 35 degrees, he got basically no difference in the distribution of the catfish within the gradient. In other words, it still kept itself near 27 degrees. In other words, it looked like 27 degrees was the preferred temperature of brown bullheads. And this was despite the fact that the brain might have been at 27 or might have been as low as 23 or might have been as high as 35. But now, so it made a good control for the trauma and, all the things, the handling. When he put the thermode in a different part of the brain, in this inferior lobe here, and back in this part of the brain, this rostral brainstem gave maximum sensitivity. Then, if he let a fluid flow through the thermode that was relatively cold, the catfish selected a warmer place in the gradient. If he let the fluid be relatively warm, the catfish selected a cooler place in the gradient. It was as if the catfish was saying, hey, my brain is saying I'm too cold or I'm too warm, and so what I'm going to do is move around until my external receptors say that things are better. So you can see that this sort of behavior would suggest that there's some sort of compensatory behavior going on in response to the differential between the temperature inside the brain, deep in the core presumably, and the superficial temperatures of the skin receptors. So, looks like, it's not just a matter of either skin temperature or deep temperature. It's a comparison of the two that fish are using to manage their behavior. He did some other experiments too, having to do with escape from compartments that were relatively warm or cool into a neutral compartment, depending on brain temperature. But I think that was the most convincing. So, in elasmobranchs and sharks, there are these devices called ampullae of Lorenzini that originally people thought were thermoreceptors, but I think that the evidence has mounted to the point where they're now considered to be primarily devices that perceive electric field distortion. And if they have any thermal sensory function, it's probably peripheral or secondary to their detection of thermal fields. Touch in fish, as in you and me, is a matter of perception of stimuli at the skin surface mostly, of the tactile sense. And the receptors that are responsible for your sense of touch seem to be the same free nerve endings that are responsible for temperature, superficial temperature detection. And so it looks like the senses of temperature and touch may be coupled in fish. And some of the experiments that would suggest that are things like if you mechanically stimulate the skin of a fish at the same time you change the temperature, it's more sensitive to the temperature change than if you don't. So touching it with a little glass rod, even though that glass rod is neither warm nor cool relative to the water, tends to enhance the fish's ability to detect the fact that temperature is changing. That's the kind of information that's been used to argue that touch and temperature senses are coupled. Fish both hear and feel vibrations in the water that we think of as being sound or vibration. If it's a high frequency vibration, then I guess we use the label sound. If it's a lower frequency vibration, at some point the thumping from the car next to you, through its open windows gets translated from sound into vibration, and you can feel it. So fish are the same way. But they use two different receptor systems to get that information. They use both the inner ear as well as the lateral line system. The maximum sensitivity to vibrations of the water seems to be in the range of about 300 to 500 cycles per second. But the range is all the way down to 16, which is definitely in the range of a low frequency vibration that you would tend to feel more than hear if you were a human, up to 5,000 cycles per second. In the fish groups, the majority of fishes that are labeled as being non-osteophysi, that's the supergroup, And the Asteriophycins, which are minnows and catfishes and fish like that, tend to be more sensitive to the high frequency end of the spectrum, all the way up to 13,000. And you can compare that to humans. We hear sounds, some people would say down to 20, but I've got 50 here, 50 to 20,000 hertz, or 20,000 cycles per, hertz is a cycle per second. And I can't hear 20,000 hertz, and the older I get, the lower the maximum frequency I can detect. You know, you go to the audiologist and they play sounds in your ears at different frequencies. Now, do you hear anything? Do you hear anything? Let me know when you hear something, you know. And when they test me, I'm down around 15 or 13 even. So older people and larger boned people, larger bodied people tend to be less sensitive to high frequency than people that are not. In fact, and women tend to be sensitive to the upper end of the frequency spectrum more than men. Peak sensitivity in humans tends to be about 2,000 hertz, 2,000 cycles per second. You know, we're designed to hear in air, and fish hear in water. So I think that accounts for the big difference in the frequency that's optimum. Fish, I think, like people, literally hear higher frequency vibrations as sounds, and the inner ear is the receptor, and you've got these semicircular canals, and they've got endolymphatic fluid that sloshes around and stimulates hair cells that line the labyrinth, and the triggering of these cells is summated and converted and sent to as auditory nerve stimulation. These things are called neuromasts, and they're concentrated in these little swellings called ampullae at ends of the semicircular canals. The ampullae of Lorenzini are related in elasmobranchs. So the inner ear looks a lot like the human inner ear and works about the same way. The Asteriophycins do things more and better than ordinary fishes because they have a device called a Weberian apparatus. And the Weberian apparatus is a series of bones derived from anterior elements of the vertebral column processes that functions like a mammalian middle ear, in that, you know, we've got the malleus and the incus and the stapes, the hammer, anvil, and stirrup that connect our eardrums, which separate the middle ear and the outer ear from the inner ear with the membranous labyrinth. And so basically the vibration of the eardrum is transmitted by this mechanical linkage of bones to the inner ear. Fish don't have that arrangement. What they have is a series of bones but they have analogous functions that connect, and they don't have outer ears, and they don't have eardrums, but they have the analogs of those devices in the case of the Asteriophycin fishes. The analog of the eardrum in mammals is the swim bladder. And the analog then of the middle ear bones of mammals is the Weberian apparatus. And then the inner ear is both analogous and homologous. It's analogous in that it functions in a similar way, and it's homologous in that it's derived in the same sort of way developmentally. So that's why we think Asteriophycins are so much more sensitive to high frequency sounds than non-Asteriophycin fishes. Fish hear these high frequency vibrations, and they feel low-frequency vibrations. And I would classify low-frequency vibrations as being ones less than about 200 cycles per second. And the anatomy that's important there is the lateral line, which I say more correctly, I guess, is the acoustico-lateralis system, the lateral acoustical system, but it's not just a system on the sides of the fish, which is where we normally see it, but it also extends onto the head. And if you look carefully at the head of a fish, like a centrarchid, you can see the little pores that lead into this head end part of the acoustico-lateralis system. The functional system is a lot like the inner ear in that there's a fluid that's sloshing, and that fluid sloshes in sympathy with the vibrations of the external medium. In this case, though, instead of being in the lymphatic fluid inside the semicircular canals, triggering neuromasts that are in the ampullae and to some extent in the canals themselves, it's just water. And the water is sloshing in the canal of the lateral line and triggering the cells that I think are, again, both analogous and homologous to the ones that are in the inner ear. At least they're called neuromasts. Little epithelial cells with little cilia, little hairs that stick out and get triggered when the water sloshes back and forth. Lots of different experiments, some of them pretty clever, have revealed the multiple functions of the lateral line system in fishes. And probably the major function is simply to detect the flow of water over the fish's body and sort of know when the environment is moving relative to the surface, so that fish can control hydrodynamics as they swim. Also, fish can use the acoustico-lateralis system as sort of a crude radar or sonar system, a crude echolocation system. And folks have done things like cut the nerves and show that fish will blunder into clear glass obstacles if they don't have the functional acoustico-lateralis system. But if they do, then they'll avoid the clear glass. And even when you blind them, they will still, or cover their eyes, they will avoid bumping into objects if they have intact lateral line systems, but not if they don't. Part of this flow of water and hydrodynamic control extends to optimizing relationships among individuals within a school of fish. And this is particularly important in obligate schoolers, ones that really are schooling fishes like herrings and tunas to a certain extent. Not so important in fish that sort of group together, out of need or convenience, but fish that orient parallel to each other, heads pointed in the same direction, and they tend to swim at distances that are related to the size of the fish and the speed of movement of the school. And that tends to be governed or managed, I guess, by the signals they're getting from their lateral line systems. And fish can detect movements of prey by the vibrations of the prey as the prey swims, and they use their acoustico-lateralis system to do that too. So all these things turn out to be important. And somewhere they should be on YouTube, but they're probably not. There are all these neat old films that people shot. I remember seeing one of a school of minnows that had been, I think, experimentally blinded, and they were in a tank, and they were taught to, they would feed depending on the vibrations of the food, and so if you touch the surface of the tank with a little, tap of a little metal or a little glass rod, the fish would come there, looking for food. I mean, they were obviously picking up the vibrations of what was happening, the movements of the water being triggered by the movement of a glass rod. Electric fields and the distortion of electric fields that are generated by the animal itself, as well as electric fields that are coming from external sources, are sensed by receptors that are derived from the lateral line system. I mentioned already the ampullae of Lorenzini in sharks. Fish use their electric sense both as a sensory device as well as in extreme cases as an attack device or a defense device. Some fishes generate electric fields that are weak and use those electric fields to act again as sort of an echolocation and to detect the presence of prey that are generating weak electric fields. And other fish have well-developed electric organs that are so strong that they actually can produce electrical shocks, as you well know. And there seems to be, as I say here, a continuum between fishes that use their electric sense more as a sensory device and those that use it more as a defensive or offensive force. Fish that have strong electric fields, I'm trying to get my notes straight in my mind. Fish that sense electric fields, those generate song fields, shared characteristics, and I guess this would be both strong and weak. But I'm thinking mainly about weak electric field fish like the gymnotiforms, the elephant fish and that group. They tend to have as common characteristics the fact that they tend to swim with their body held pretty much rigidly as opposed to the serpentine motion that you think of as normal for fish. And in fact, a lot of them have elongate anal or dorsal fins, and they move along by just rippling those fins. And they tend to be fish that live in murky water. Usually they're freshwater, although not always. There's some good cases of strongly electric fish, like electric eels, or like electric rays, I should say, that live in saltwater. They move with these elongated median fins that I mentioned, and they have enlarged cerebellums of their brains that seem to be associated with this electric sense. So those are things that tend to be in common. And, extreme electric fishes that can generate shocks have electric organs that have different developmental origins. They're probably polyphyletic in their origins. And there are four families of fishes that are strongly electric, and they're the rays, the electric catfish, electric eel, and the stargazer. Electric organs consist of flattened cells with a nerve interface on one side and arranged to allow electrical summation, so you can get a big charge like a capacitor when it discharges. And I'm thinking, and I may be wrong, but I think that the stargazer's electric organs are derived from neural tissue and all the others are muscular. So, that sort of thing leads you to think they came from different places. No. The gymnotiforms are derived from neural tissue. I've got it here. Electric organs are mostly derived from striated muscle, but one South American gymnotiform at least has that tissue from neural tissue. I always like to point out at this point that I wish somebody would really get in and look closely at that ancient fish in North American waters called bowfin, Amia calva, or grinnell as I knew it growing up, because it's got a lot of these characteristics superficially that would indicate that it has an electric sense and uses it. It's got that elongate, rigid body. It's got elongate fins, and they ripple when it moves. It lives in murky water. It's got little bitty eyes. I don't think they see very well. And I just wonder if somebody shouldn't get in and look at the cerebellum, for example, of bowfin to see if, or maybe do some experiments to see if they have any electric sense. But as far as I know, there's never been any publication on that. Chemoreception. So in the case of vibrations in the water, there are two sets of sense organs that have some common features involved. There's the inner ear and there's the lateral line system. Vibrations also, the chemo senses of fish also have two sets of organs involved, just like ours. There's taste and smell. And it looks like, taste is sort of the crude sense, and smell is the refined sense, much more sensitive, much more distinguishing. Taste, or I guess some people use this fancy word, gustation, involves taste buds, epithelial cells that are mainly in the mouth, but actually inside the fish as well as outside. They go back into the pharynx. They occur on the lips. They tend to be concentrated in the barbels, if there are barbels, like catfishes. And even on the skin of the head, the external surface can have taste buds. These taste buds allow fish to discriminate the same four basic taste sensations that mammals are known to distinguish. At least my information indicates. I'm not sure if there's anything more recent, but they're, the sweet, sour, salty, and bitter are the four senses that we tend to report from. If somebody holds their nose and tastes something, they usually can distinguish these four things. The sense of smell, or olfaction, seems to be much more refined and much more acute, and it's much more acute apparently in fishes than it is in mammals. The receptors are specialized epithelial cells that line the nasal capsule, and in forms like eels that are especially sensitive to smells, the sensory epithelium is folded and refolded into what's called an olfactory rosette. So lots of surface area with lots of receptors. Some fish have a two-way access to, in other words, their olfactory capsule is a blind sac. Fish like sculpins and sticklebacks have only a single nostril on either side, or nares. And then there are other fishes that have incurrent and excurrent nares on the same side. The flow is one way then through the olfactory capsule and over the olfactory rosette. Most minnows have this kind of an arrangement, for example, and some even have a little flap of tissue on the posterior side of the incurrent nare that sort of directs the flow of water down and into the chamber. What do fish use their chemoreceptors for? Well, just about everything you can think of. The logical first guess is food finding. It's well known through conditioning experiments that fish can detect and resolve the distinctions between different kinds of potential food, even related forms of invertebrates, like, the difference between, I'm not sure if it would extend to the differences at a generic level between different cladocerans, but it certainly extends to the difference between cladocerans, for example, and copepods. So you can train minnows to react to copepods as food and to not react to cladocerans, and then do the reverse training, and fish will show you that they can detect. Plug the nares in sharks and they can't find food. Open it up and they do. I'm not sure that all those experiments that involve plugging nares really are good, well-controlled experiments that make you feel like, well, okay, yeah, they covered all the bases there. In other words, when you plug something's nose, it may just have sort of a sensory trauma response and not behave. I'm not sure that it shows for sure or proves the case that the nose is important in food finding, but it probably is. You know, there are all kinds of examples of sharks trailing a source of, you know, read The Old Man and the Sea, for example, and I think that's probably a fairly honest report. Sharks' ability to zigzag across the trail of a potential prey item and follow it to its source. I don't doubt that happens. Clever experiments have shown, very clever, some of the most clever experiments done, I think, in the fish world have been done in association with research on orientation, behavior, and homing in fishes that at home. Olfactory memory, it turns out, is very important in the stream phase, particularly of homing in salmon. But even in fishes like sunfish that don't do a lot of homing, it's clear that they depend partly on olfaction to relocate home once they've been displaced from home by either natural or unnatural means. A guy named Gunning one time took sunfish and displaced them by moving them up or downstream from their original home site, and showed quite conclusively that the fish that were displaced downstream had a much greater chance of returning home than the fish that were displaced upstream. So, you know, what's different between downstream and upstream? Well, if you're downstream of home, then the essence of home is being carried in the flow of water downstream to where you are now, and all you have to do is exhibit positive rheotaxis, swim into the flow when you detect home, and not exhibit positive rheotaxis, either just behave neutrally or actually do reverse or negative neotaxis if you don't detect home. And it looks like that's the mechanism that salmon use in the homing phase of migration, in making choices at the points of confluence in the stream system. In other words, just swim as long as you detect home. Swim upstream. If you quit detecting home, then just turn loose and drift. And as soon as you detect home again, then turn on the swimming and keep going. And so the chances are, you know, that if you come to a fork in the stream, you're downstream from the confluence, you're detecting home, you make the wrong choice, you choose the right fork is not home. So you just stop swimming and start drifting. And then you try again. And eventually you get the left fork. And of course you get stronger home there than you had before because it's a little more concentrated, and so you keep swimming more strongly. So it looks like these are the kinds of mechanisms in the way olfaction operates to allow those to work. You know, I was associated early on peripherally, I'd say, not in any kind of important role, but just as an observer in some of that early olfaction research that was done at Wisconsin in Arthur Hassler's lab there. And worked closely with a fellow graduate student named Andy Deason. And Andy and I subsequently went together and worked with the National Marine Fisheries Service in Hawaii. Andy's still with the Fisheries Service, I think, in San Diego now. Maybe he's retired. But one of the things that Andy did was to imprint juvenile coho salmon on artificial olfactorants. And so he took groups of fish that had never been exposed to this unnatural substance, is actually an alcohol. And he would drip this morpholine was the substance into the raceway where the treatment group was kept, and then drip just regular water from the raceway into the other control group's raceways. And then rack them, as we called it, put them in a perfusion trough under some local, under probably the technical equivalent of curare, which paralyzes the motor system. I'm trying to remember the chemical name for curare. Does anybody know? Anybody ever work with it? A trimethyl something or other. But anyhow, so the fish that had been conditioned, had been imprinted on morpholine, were then kept for a long time without exposure to the morpholine. And then he would rack them, both groups, and what he showed was that the electroencephalograms from fish that had been imprinted on morpholine were different from those that had not been. And then subsequently, folks used morpholine to imprint fish to an artificial stream where there'd never been a population before. These are freshwater fish in the Lake Michigan system, coho salmon, and showed that you could generate a run. That is, you could generate a subpopulation that would home to that stream just by dripping morpholine into the water. And they would come back there and spawn then. So it was a way to create new spawning subpopulations. Pretty elegant stuff. Another thing that has generated a lot of interest in the area of chemoreception is the fright reaction that is exhibited by certain osteophysins, particularly minnows. It's a fellow named von Frisch, a German, did the original early research on this. And, of course, he used the German words to characterize it. He called it a Schreckreaktion. And Schreckstoff was the fright substance. And his work showed that if you damage skin cells of some minnows, substances are released into the water that cause a fright reaction in conspecifics. And that reaction is also somewhat effective even in more distantly related species. It sort of progressively becomes less effective as you move taxonomically away from the fish that are siblings, the actual conspecifics. And it turns out that there are specialized cells, club cells in the skin of these osteophysins that almost look like they were designed to be fragile, so that if there is any kind of abrasion, if a predator attacks and breaks the cells, then these substances are released into the water, and it looks like the functional relationship is that conspecifics are warned by the presence of the fright substances in the water that there's danger, and so they will go into a series of stereotypical behaviors. In the case of some, it amounts to forming sort of a ball, a densely aggregated group, and the interpretation of that kind of behavior is that that's a good defense mechanism on the part of schooling fish, because the group packed densely together sort of looks like a super organism, a bigger fish to a predator, and maybe would prevent the predator from attacking. We tried to do, before your time in biology of fishes, Ray, we tried to do Schreckstoff experiments in biology of fishes, but one thing we learned was that, you have to have a very optimum situation. You have to hold your mouth right and be real quiet and not make the fish nervous for any reason if you hope to demonstrate anything when you do this. Otherwise, they're already panicked before they get this fright reaction. So the students are enough to cause a fright reaction. So we gave up on that. It just didn't work. And I mentioned semiochemical signaling between conspecifics here, Ray, and I introduced that based on work we've done together. My colleague here, Ray Camps, published a paper in Chemical Ecology some years ago arguing that animals may send materials away that depend on the physiological state or the general welfare and health of the generating animal that could control the behavior of conspecifics. For example, if you were in a world where there was too many of you and density was too high to be optimum, one way to manage the future from your own point of view would be to generate a semiochemical that would repel conspecifics. Or, better yet, tell conspecifics, hey, don't think about reproducing. There's already plenty of us around. And, if you're a larval form or a juvenile, don't metamorphose because we already got plenty of adults. And so we were thinking about this in the context of aquaculture and the possibility that these semiochemicals might be released by high aggregations, high densities of shrimp. Because crustaceans are the world's best communicators when it comes to producing pheromones and substances, that have a... So, there's a lot of literature, and Ray reviewed it, not so much with fish, but in things like rats, where rats under stress generate stuff that keeps other rats at bay, maybe even makes them suicidal, interferes with reproduction. So what else, Ray? Have you learned anything new that you can add to that story? Did I tell it pretty well?
Yeah, I don't remember the rats. I thought we pretty much stuck to aquatic stuff, but we did look at snails, frogs.
Snails, frogs, Ray says. He doesn't remember the rats. How about the lemmings? Did we talk about lemmings?
What's that?
Did we talk about lemmings jumping off the cliff? No.
We talked about lemmings, I don't believe.
Yeah, so, these maybe... It was pretty speculative, and I don't think anybody's just been picking up that ball and running with it from what I can see in the literature.
We've been cited.
Yeah, but... So, it's the kind of thing you ought to worry about if you're in aquaculture, because if it's possible that if you're doing something that's so artificial that you have to, as you have to do it when you're doing intensive aquaculture, you've got an artificial situation, right? It's going to have to be. And if you discharge effluent, and if that effluent is carrying signals downstream, you hope that there's not a lot of impact on the natural community that's downstream, because you don't own that one. That's the public's resource, not yours. And so I think maybe the idea that there should be zero discharge, maybe not so dumb. It's a conservative thing. It may not be essential, but it probably is good protection. So these are kind of out there beyond where we are and talking about what might be. Water quality sensing, some people like Andy Deason used to argue that fish use olfaction to detect the presence of contaminants and avoid places where contaminants are concentrated, and that some minnows can detect and avoid chlorophenols at concentrations that are below back in those days you could detect with the best chemical methods in the analytical lab with gas chromatography. Salmon can detect and avoid copper as low as 9 parts per billion, which is only 2/10 of the incipient lethal level. So that's good. You wouldn't want to detect what was 2 times the lethal level. You'd already be dead. So it's nice to be able to detect something that's sublethal. And I don't know if this is in my notes or not. I don't guess it is, but I read someplace that somebody calculated that eels in their olfactory rosette could detect concentrations of potentially lethal stuff where there's only one molecule of the stuff in the in the capsule at a time. But then somebody else said, yeah, well, that assumes that you're just taking a flow of the water and that the stuff is concentrated at whatever you're measuring analytically. It assumes there isn't any kind of active absorption or grabbing of these molecules to amplify their signal. So I don't know, but it's pretty amazing just the same. I think that all who work in this area would agree that aquatic organisms, fish, are very sensitive to dissolved materials. So vision looks just like a human eye. Well, maybe not quite. You know, it's got a lot of the same stuff, right, that you'd see on the eye chart if you went to your optometrist or ophthalmologist. You know, it's got a cornea, it's got a lens, it's got a retina, it's got an optic nerve, it's got a vitreous humor behind the lens, and it's got an aqueous humor in front between the lens and the cornea. It's got an iris. This lens, though, is a little different. The lens, in the case of most fishes, is a pretty rigid structure. In fact, it's often about all that's left in the stomach of some predatory fish. It's a little spherical, hard, translucent thing, not real clear, typically by the time we see it at least. That's not like the lens in a mammalian eye, which is much more flexible and usually sort of a biconcave structure rather than a spheroid. So what else is there? There choroid coat, guanine crystals to reflect light. A choroid space, vascular, nourishes the retina, has an oxygen-concentrated choroid reap that I already mentioned, that except in elasmobranchs, and sometimes you get a bubble forming in supersaturation conditions that cause pop-eye in that choroid reap. Sometimes pop-eye is caused by other things like parasitic infection. The iris is fixed in teleosts except flatfishes and eels. These are notes that I'd forgotten I even wrote on there. So, vision, distance accommodation, you know, that's the maneuvering of the eyeball and its contents to accommodate for objects to be seen at different distances from the animal. And in the case of humans, you know, especially young humans, younger than I am, most distance accommodation is accomplished by changing the shape of the lens itself, making it fatter when you want to have a short focal length and thinner, more like a plane when the focal length is long. And there may be some lens shape change in sharks that's analogous to what goes on in mammals. But teleosts mostly accommodate for object to eye distances that are variable by moving the rigid lens back and forth inside the eye, moving it toward and away from the retina to change the focal length, pretty much as you do with a telescope. You know, because you don't have a flexible lens in a telescope for the most part. Trout have the ability to focus simultaneously on nearby objects and far objects. Well, everything is relative. When I say nearby, I mean within striking distance. If it's food, you know, like less than a fish length, maybe a head length away, versus maybe a couple of meters away. And the way they do that is they have instead of having a spheroid, they have more of an ellipsoidal lens that's oriented sort of along the axis of the body of the fish. Long axis head, short axis to the side, so that when things are seen in front, like food right in front of the snout, then the focus is up close, which is as it should be, and then something that's out here, like a predator that's sneaking up in the side, is seen further away in sharp focus, or as sharp as the focus gets with fish. My suspicion is that fish, you know, I think the evidence is that fish don't have anything like the visual acuity that mammals have. In fact, I may even have some data in here. Yeah, maybe something. Sooner or later, I got something in here. Fish have differentiated photoreceptors, in at least the teleosts that people have mostly studied. They have cone cells that are designed to function at relatively high light intensities, and then rod cells that are more able to see in low light levels. And the separation tends to be about 10 to the minus 1 foot candles, and as I recall, that's about like a new moon night, or maybe a little bit brighter than that. In bright light, the rod cells are protected from overstimulation by actually migrating into the pigment layer of the retina. Diurnally active fish from brightly lighted habitats tend to have more cone cells. And then deep sea forms and fish from deeper in the water tend to have more rod cells. And even in the really deep forms, the rod cells have convergent fibers that converge, that summate the information coming from multiple cells on one optic fiber, so that you get this electrical summation, basically, make them able to see better at low light, really light light. And there's some stuff here on visual pigments that given up on making any sense of, and I think the literature seems to have given up on it too. I don't see much reference to these things anymore, but it has to do with the fact that fish have two types of pigments, rhodopsin and porphyropsin, and they have different peak sensitivities, and apparently they're designed to allow optimization of whatever the color spectrum is in the habitat where you live. But when you look at all the logic involved, I'm not sure it makes really good sense. Many freshwater and euryhaline fishes have both types of pigments. Freshwater fish that have only one pigment tend to have porphyropsin. And some people have tried to make a deal out of the fact that freshwater habitats are murkier and usually light penetration's not, I don't know. Marine fish that have only one type tend to have rhodopsin. But a lot of freshwater fish have both. A lot of marine fish have both. Color vision definitely occurs in some diurnally active fish that live in brightly lighted habitats. And just think coral reef, and think about butterfly fishes and parrot fishes, and things that have bright colors tend to see color. Things that don't, like tunas, for example, even though they look brightly colored to us, they're mostly just contrast studies, I think, to other tunas. And they don't tend to have color vision. They have the ones that do see color have the same three pigment system that humans have. Red, green, and blue tends to be the maximum sensitivities at these wavelengths. Yeah, I did say something about visual acuity. 6 to 11 minutes of arc tends to be kind of standard for well-lighted conditions. And how that's measured, by the way, here's what visual acuity means. It means that, imagine two marks on the wall, and think about the angle between the first mark and your lens and the other mark. If that angle in your fish is more than about 11 minutes, then you can resolve the fact that there are two marks. But if the angle is less than 11 or 6 or 11 minutes, then it looks like just one blurred wide mark. That's what the idea of acuity is. And, of course, it depends on how lighted. That's under well-lighted conditions. Is that making sense? I see Zoe sort of squinting her eyes there. So I'm going to put marks on the board for benefit of Zoe here in the room. And I realize you're not going to be able to see those in Corpus or Palacios or any place like that. But so it'll be hard for me to put two marks up here that Zoe, only sitting 20 feet away, can't see, or in fact 2 marks. Can you see that there are 2 there, or does it just look like 1? See, she's got so much visual acuity. You probably got seconds of acuity instead of 6 to 11 minutes. Fish, a minute is a 60th of a degree, and a second is a 60th of a minute. So, I don't know, visual acuity of humans, I think, is down in the seconds. How about you, Valdemar? Can you see 2 marks there? How about Nell? Can you see 2 marks? No, I faked you out. I just made 1 mark. That's not You can pull those little tricks. They don't mean anything. They're fun. Okay, so fish do not have as much visual acuity as humans. They can discriminate geometric shapes, and I'm not sure if we know what the limits of the geometric shape discrimination are, but fish have been trained using positive reinforcement, the same kind of Pavlov thing that we talked about, to discriminate between circles and triangles, for example, of the same area or the same linear dimensions. Even to discriminate between a triangle pointing up with the apex up, one apex pointing up versus that apex pointing down, which is pretty neat. I don't know if they can discriminate between 5-pointed stars and 6-pointed stars. Maybe. One time I got interested in whether or not they could distinguish between a little English character C and W. Why would I be interested in that? Well, I'll get to that story later, but it has to do with predictive regulation of environment and being trained to recognize C meaning cooler and W meaning warmer. But I never did do that experiment, Ray. We'll talk about predictive regulation of environment, though, in a bit. Some fish have binocular vision, and it's just like with warm-blooded animals, if the eyes are both in front, they tend to be binocular, but if they're to the sides, then probably not, Pikes have sighting grooves on the snout. The Esox, the Muskellunge, northern pike, you can actually see those grooves in the bone. And it looks like the angle between the two eyes and using the sighting grooves, you draw lines from the two eyes that the crossing point is just about the distance that would normally be the prey strike distance. So they're designed to be able to look at the prey right in front of their snouts at that distance. So the final thing I'll talk about has to do with one of my standard discussion questions, Chris, on the final exam, which is the visual window of fishes and how it works. And it has to do with the fact that light is refracted when it passes from the air into the water, or from the water into the air. And so a fish that's sitting here down in a clear, calm water with no ripple on the surface or anything, very clear, sees the whole world above the water through an angle that turns out to be 97.6 degrees. And that's a pure property of physics of refraction in light and water. And the reason it sees the world through that angle is that light rays that come in at almost flat to the surface, normal to the surface, parallel to the surface, are maximally refracted by that much. Any light ray that comes in from here to here or from over here to over here is refracted down, and the fish can see that object. It's called a fisheye lens, right? It's a wide-angle lens. And so the whole above-surface world is compressed into an angle of 97.6 degrees as far as the fish is concerned, or as far as a human diver is concerned. This has nothing to do with being a fish. It's got to do with the physics of water. Go down in the water, look up, you see a circle of light, and you probably don't see a lot of clarity of objects above that surface because it's pretty distorted, but that's the angle of the cone when you look up is 97.6 degrees. So here I've attempted to draw what the fish sees when it looks up, and right in the center of the visual window is the sun that's directly overhead because it's not refracted. The only light ray that's not refracted is the one that's coming straight in, perpendicular normal. That's the normal, right? I said that word wrong before. The normal line here is the one that is perpendicular to the plane. And this bird that's over here off to one edge, it looks like it's closer here because it's actually seen along that line. It's seen here where my pointer is. In other words, if you extend that line straight up from where I've got the little arrow there. It's seen there. The tree, the tip of the tree, is seen to be, out here somewhere. So right there. So what about the rest of the world? What about all the world that's the underwater? Well, it's seen as a reflection. If it's seen at all, it's either seen directly for where it is without any refraction because it's still inside the medium, or it's seen as a reflection from the surface, off the surface. And there it's the angle of incidence equals the angle of reflection type rule from optics. So, you could draw me a picture of the visual window of a fish, and you could say that what it means is the 180-degree world above the surface is folded by refraction, not reflection, refraction of light into a visual window, making an angle from edge to fish and back to the opposite edge, an angle of 97.6 degrees. And if you told me 95 degrees or 99, I'd probably say that's close enough. 96.7 if you got those two digits, if you couldn't remember which, that wouldn't be that important to me. I've tried to stress all along in here, it's mostly the concepts that I'm trying to get you to engage, and not so much the specifics or the numbers or the little details like that. But I think you can see the concept is pretty important. If you don't think the visual window is important, have you ever tried to bow hunt fish, or spear them? If they're right on the surface and you can actually see a little bit of fish above the water, it's no problem. But if they're deeper than that, you've got to learn to compensate for the refraction of the light ray that's coming from the fish, which is what you're seeing, through the water into your eyeball. And so, if you were a person there instead of a pine tree and you were trying to spear that fish, you'd be aiming at a point right there. And your spear would be not refracted. It would just go straight on. It wouldn't go very much because water's a pretty good, water impedes things. You shoot a high-powered rifle into water. You don't have to have very much water to stop the bullet. There's a lot of resistance there. But if you could shoot, you would shoot along the line where you saw the fish, and where you saw the fish would put this arrow out here someplace above. You overshoot every time. So if you're a bow hunter, a fish spearer, you got to compensate by intentionally taking into account the refraction and aim below. And similarly, the fish has to aim, it has to aim in compensation for refraction too. And some fish are really good at that. Why would you ever care? Well, I saw a demonstration once, and it had to do with archer fish. And Ray's probably heard this story. I had a technician one time who had a little archer fish as a pet in an aquarium. And archer fish have learned evolutionarily that they better be able to compensate for refraction, or else they can't spit that drop of water out and hit a little insect that's sitting up there on that piece of vegetation above the surface. Because if they don't take into account the refraction, they're going to shoot too low or too high, whatever it is. I guess they'd shoot too high. But they're pretty good at making that adjustment. And this particular archer fish was the original anti-smoking archer fish, because it was fond of putting out cigarettes. Anytime anybody would hold a lighted cigarette over the tank, it would spit and put the cigarette out. I don't know if it was trained to do that or if it just didn't like smokers. Back in those days, lots of us smoked. So with that story, do I have something else to say? Yeah, I've got a few more things to say that I need to say here, and I sure would like to say them, but you know what? I'm not going to keep you, so we're going to finish this up. It's 12:25 in College Station. We are in good shape as far as getting coverage of the material that I wanted to cover during the semester. I think we'll finish in good style. So I'll just stop here.